Hello students and welcome back to our channel. This is Assistant Professor Miss Karuna Komatwar from Department of Biotechnology, Rajarshi Shahu Mahavidyalya. So today in this video lecture, we are going to study about the topic entitled as Elisa. Moving forward to the definition, if we just look deep into the definition, what is the full form of ELISA? ELISA means it is an enzyme-linked immunosorbent assay, which means it is a technique which is specifically used to measure the concentration of either antibody or antigen present inside a body. When we are speaking about antibody, antibody means what? It is something which is present naturally inside our body, and when a particular outside material, which we call as a foreign material, any dust particle we can say, for the very best example, if a dust particle gets inserted inside our body, it gets induced inside our body, then what happens is our body has a natural defense system. Means what it does is, अपनी बॉडी के पास एक एबिलिटी होती है कि हमारे बॉडी के अंदर बाहर से कोई भी अगर कोई एक्सटर्नल पार्टिकल आए विच इज हार्मफुल फॉर आवर बॉडी, देन आवर बॉडी व्हाट इट डज इज, हमारे बॉडी क्या करती है, उस पर्टिकुलर हार्मफुल एजेंट को किल करने की कोशिश करती है, अल्टीमेटली किल कर देती है, which is called as antibodies. तो अपनी बॉडी क्या करती है, एंटीबॉडी प्रोडक्शन करती है, with the help of which the outside material gets destroyed. And when we are speaking about antigen, antigen means what? It is something which is getting inserted inside our body from outside material or outside surrounding. So this is the definition. Moving forward, if you just break down this word enzyme linked immunosorbent assay, what it means? Sorbent means what? As you can see already on the screen, it is an antigen or antibody of interest that is absorbed on plastic surface. Now plastic surface means what? Here when we will be seeing in detail about the types of ELISA, there are basically 4 types of ELISA, and for that purpose what happens is you require this micro titer plate. And what does this micro titer plate do? You are adding different types of solutions, and after adding the solution, what happens is, if you are, for example, if you are adding either antibody or if you are adding either antigen, these solutions that are getting binded, for example, this is a micro titer plate, this is one of the well, for example, just a second, this is your well, and what happens inside a well, if you are adding antigens, specifically antigens are getting attached to this surface, that is called as what? Sorbent. What is happening? This particular antigen or antibody or any type of whatever the molecules you are adding, that is being added or absorbed on the surface. This is called as sorbent. Coming to next, immuno. See, from the name itself, what happens is any outside material that is being recognized by our body, and our body or leads to the production of specific type of antibody. You can call antibody as a soldier, and you can call antigen as a thief, right, of our body. So immuno means what? This particular thief, it is recognized by specific soldier which is present inside our body. Coming to the next term, that is enzyme linked. Now what does this mean? When we will be seeing in detail about the types of antibodies, which will be of 4 types basically, there is an antibody to which at a certain portion of this antibody, what is being attached? An enzyme is attached, right? That is called as enzyme linked. You can read this. This antibody is recognized by second antibody which has been enzyme attached, which is called as enzyme linked. We'll be discussing this later. Lastly, substrate reacts with enzyme. Now, however, we have already said here that on any of the surface of the antibody, not basically any of the surface, at a particular portion of antibody, enzyme is linked. And if enzyme is linked, what is the further process? We already know the protocol that enzyme, it is getting binded with substrate to form a product, right? Simultaneously, after addition or linking of this enzyme with the particular type of antibody, a substrate is added, and this substrate, it is having the ability to produce the product, which is going to display in the form of colors. Moving forward, when we are going to deal about different types of antibodies, before studying that, we have to know about basic terms of the thing which we are going to use in the types of ELISA. What is the first basic term? This is the solid phase. As I have said you earlier that this is the micro titer plate, means basically you can tell it as well plate. And in this well, what you are adding? You are adding the solutions. It may be patient serum sample, it may be antigen sample, it may be any of the sample which we'll be discussing later. So this is the micro titer plate, and which is called as solid phase. The solid phase, as you can see, there are 8 into 12 well format, means this is one sample of the well. We see there are different types of micro titer plates which will be having more than this wells also. So this is just for diagrammatic representation. Moving forward, as we have already discussed about the term sorbent, which is called as adsorption. Now, adsorption means what? From the name itself you can analyze something which is getting attached on the surface of a particular thing, that is called as what? Adsorption. For example, the process of adding an antigen or antibody diluted in buffer, so it attaches to the solid phase on incubation. What happens in this case is either antigen or antibody, when you are adding in the microtiter well plate, in that particular well, either antigen gets attached or either antibody gets attached. This is called as what? Adsorption process. Adsorb means on the surface it is what? It is attached. It is not absorbed, it is attached. What is the next thing? Washing. See, there are some unwanted particles while you are performing this different types of ELISA. For removing that unwanted particles, you are doing the washing step at each and every phase. What happens with the help of this? The extra unwanted material which you don't need in the process of ELISA, that you will, what you do, you will elute out with the help of this washing phase. And in this washing phase, what you are using? You will be using either buffer solutions, varieties of buffer solutions, right? This is about washing. The simple flooding and emptying of wells with a buffered solution to separate bound from unbound reagents in ELISA. If you think that antigen has been attached with antibody, so further what will happen? It will form a complex with antigen and antibody. And after that, what happens is the extra antigens or extra antibodies which are free in nature. Free means what? In a solution, they are neither bounded to each other, neither they are bounded to any of the other molecule. That unbounded materials you have to elute out or discard. For that purpose, you are using what? Solutions. It can be called as buffer solutions. What is the next basic term? Antigen. Any molecule that elicits the production of antibodies when it is introduced into the body. What our body is having the functionality, whatever the harmful particles are getting inserted in our body, it is having the ability to throw them out. This is called as what? Antigen. This is something which is harmful to our body. In sometimes it is not harmful, that is a type of antigen. We can also call it a small antigen. So that is not a point of this particular thing. So we'll not be discussing about it. Our point is something which is harmful or which is entering inside our body from the surrounding. Antibodies, these are protein molecules which is produced in response to antigenic stimuli. Antigenic stimuli means what? When an antigen solution enters inside our body, then what happens is it is producing some signals, and that signals are being detected by the antibodies which are present inside our bodies. Not exactly antibodies, there are some cells which are identifying the external materials signals, and that cells will have the ability to produce different types of antibodies, either different types or specific type of antibody. Because if you want to kill a particular pathogen, you need to develop a particular type of, what we say, if in normal life, if you are like suffering from fever, or you are going to take a particular type of tablet which is going to cure your fever, right? Simultaneously, if a particular antigen is getting inside your body, it is producing signals. Our body, the cells of our body, what they are doing, they are analyzing this particular stimulus, and according to this stimulus, our cells are going to prepare a specific type of antibody, and this antibody will attach on its surface, and they will destroy this antigenic molecules. Next is enzyme conjugated. As we have discussed, based upon the full form, that was called as enzyme linked. Here, an enzyme that is attached irreversibly to an antibody means what? on the surface of the antibodies somewhere at a particular portion, which is called as, basically you can call it as Fc region. At this Fc region, what happens is this enzyme is linked. And what do you mean by the term irreversibly? Irreversibly means what? You can never detach that enzyme from the antibody. That is called as irreversible, right? For example, a lot of varieties of enzymes are used in the process of types of ELISA. But for your general information, horseradish peroxidase enzyme is used basically, right? Moving forward, next your term is called as chromosome. Now chromosome, you can also call it as substrate. Now you already know what does a substrate do. If there is an enzyme, this substrate, it is getting binded with that particular enzyme, and further product is formed here, with the help of which you will be able to see some type of color in the solution, which represents that the enzyme has been bounded with the substrate and further product has formed. The display of the color in the well denotes the presence of the formation of the product. So chromosome, what is it? It is a chemical reagent or chemical molecule which alters color as a result of an enzyme interaction with the substrate, right? Basically, trimethylbenzidine, it is a substrate. When it reacts with horseradish peroxidase enzyme, it tends to form a product which you will be able to see with the help of the coloration of the solution. Next is stopping process. Now stopping, from the name itself you can analyze, it is a process. If you your desired result has been observed already, in that condition you are going to do the stopping process. It is a process of stopping the action of an enzyme on a substrate. If you think that this much amount of product is sufficient, in that condition you have to stop the reaction, you have to stop providing the substrate content. So for that purpose you are calling this process as stopping process. Next, reading. After the display of colorants, you have to observe, you have to take the readings, and with the help of which equipment? Spectrophotometer. With the help of this you will observe the readings. Spectrophotometric measurement of color is developed in ELISA. With the help of this you will be able to note down the readings. So moving to our main content, what are the advantages of this ELISA? ELISA are having basically majorly two advantages. What are they? Either it is having a qualitative estimation analysis as well as it is having quantitative estimation analysis. Now qualitative means what? Here you are only observing whether a particular antigen is present inside a body or not, or whether an antibody is present inside a body or not. This is your qualitative estimation. Moving to quantitative estimation. Quantitative estimation means in this process what you are doing is you are measuring how much amount of antigens or how much amount of antibodies are present inside the patient's serum sample. That is termed as quantitative estimation. So with the help of the technique of ELISA, you will come to know whether a pathogen is present inside a body or not, or how much amount of pathogen, how much concentration of pathogen is present inside a body. As you can see here, this is the basic principle, basic diagram of the principle which is representing for ELISA technique, basic ELISA technique. Here, this particular shape, it is representing as a well. On that well, what you have done, an antibody is coated on the well. As we have seen earlier about the adsorption process, this is the adsorption process, this much. This particular antibody is coated inside a well. Further, if there is the presence of antibody, then what we need to add? We need to add antigen. Here you can see antigen is added. Now after addition of antigen, what you are adding? You are adding secondary antibody. This particular antibody which is linked with the enzyme, which is enzyme conjugated, it is called as secondary antibody. Secondary antibody is added. Now since there is a presence of particular enzyme here, then what is the next step? The next step is addition of substrate, and after addition of substrate, this particular substrate, it is getting binded with enzyme, and further it is having the ability to form the product, and with the help of which you will be able to see the color formation inside a particular well. Moving forward, not only for this, you will also be able to identify different types of diseases present inside the body. Like for example, if we take the example of AIDS, there are certain, what we say, like certain pathogens which are leading the formation of AIDS inside a human being. That pathogen is present inside a body or not in a patient, we will be able to identify the particular presence of the AIDS inside a human body or not. Moving forward, see, before going into the all types of antibodies, these are some terms you also have to know, because if these basic terms are not clear to you, you will not be able to understand what particular portions of the antibody means. This is the whole structure of antibody. As you can see here, there are light chains, there are heavy chains, as you can see here. As I have said you earlier, enzyme is bound at which portion? This is called as Fc portion or Fc domain, means what? Fragment of crystallization, it is called as, right? Additionally, this particular portion, which is called as paratope. Paratope means what? It is also called as a portion which is specifically designed to get bounded with an antigen, right? These are paratopes. Further, you can see here this portion, it is called as antigen. This is a foreign material or pathogen, right? Now this is antigen, and this antigen on its surface, it is having several portions which are called as epitopes, means that are the binding portion for this particular paratopes. Now coming actually to the types of ELISA, there are basically 4 types of ELISA: direct ELISA, indirect ELISA, sandwich ELISA, and competitive ELISA. These are the basic structures. Whatever the thing is happening inside the well, it is explained in this manner. So we will be discussing each and every term in detail. As you can see here, direct ELISA means what? Directly you are adding samples, and no any longer process is there, a very small process is there, and with the help of that process you will be able to identify whether in the particular patient sample an antigen is present or not. From this you will be able to identify where there is a presence of antigen or not in a patient sample, right? It uses the method of directly labeling an antibody itself. Microwell plates were counted with a sample containing the target antigen, and binding of a labeled antibody is quantified by calorimetry. We'll just try to understand this particular type of ELISA with the help of this diagrammatic representation. What is happening here? As you can see, this is a well, like we have discussed earlier, the microtiter plate, it is having a well. This is your well, and in this well, you have added patient's serum sample. As you can see here, sample is added to the wells, right? And in this particular thing, there are different types of antigens, right? And this also red type of antigen. So what is your, from among all these pathogens, what is your desired pathogen? Assume this red one is your desired pathogen. After adding, what you are doing is directly you are adding secondary antibody. The antibody which is linked with enzyme, that is called as secondary antibody. You are adding secondary antibody. Horseradish peroxidase enzyme conjugated antibody binds immobilized analytes. These are your analytes, and you are adding secondary antibodies. If this particular secondary antibody finds that a particular antigen is present inside it or not, in that condition what happens? It gets binded to it and it forms a complex. After that, what happens is, since there is a presence of enzyme, then what is your basically next step? That is the step of addition. And in the step of addition, you are adding here substrate, and this substrate, it is getting converted into colored product, right? And based on this coloration, we will be able to identify if there is a presence of your desired antigen or not, right? If there was no any color display, means what happened? If this was also silver in color, and after addition of this particular secondary antibody, it wouldn't have bounded to silver color antigen. And further, there will be a washing step, and after this washing step, this unbounded secondary material also would be get washed off. And after washing it, even if you add substrate, since it was already washed off, even if you add substrate, there will be no any binding, no any product formation, and no any display of coloration, right? With the help of this, you will be able to find a particular pathogen or antigen is present inside a body or not. Next is, based on this, I want to explain you that, for example, HIV virus has infected this particular human body, and because of this, what has happened? AIDS has been occurred inside this particular human being's body. So for that purpose, as I have already said, what is this virus? This virus is a thief, right? And what it does? It is going to affect, but apart from affecting, it is also having one ability that it induces soldiers of our body, which are called as what? Antibodies of our body. If like what happens is soldiers inside our body also gets developed, and after developing, what happens? You are taking the serum, like blood sample after you are performing the process of centrifugation, and with the help of this centrifugation, you will be able to isolate or separate out the serum sample from this patient, right? This is the serum sample, right? And the upside portion, which is transparent in nature, it is consisting of what? These types of antibodies, which are specifically produced in response to the stimulus of this HIV virus. So from this figure, I only want to explain that. Now, in the second type of ELISA, here we are going to discuss that a particular type of antibody is present inside a human body or not. Moving forward to next type, that is called as indirect ELISA. We'll go directly to the diagrammatic representation, with the help of which you will be able to identify how an indirect ELISA method is carried out, right? As you can see here, it is a particular technique which is used to detect a particular type of antibody against a known antigen, right? As we have discussed earlier from that image. What happens is, this is your microtiter plate, one well of the microtiter plate. And if you want to detect an antibody, then what needs to be added? Opposite of that, means if you want to detect a soldier, whether it is present inside a body or not, you need to add a thief, which is called as an antigen. So in this particular well, what you are adding? You are adding antigens. These yellow circles are termed as antigens. Okay. After this, what you are adding is, you are adding the patient's serum sample, with the help of which only you will be able to analyze whether this particular antibody is present or not, right? You will be adding patient serum sample. If that patient serum sample, it is having a specific type of antibody, then it will get bounded with this particular type of antigen. If it is not having a specific type of antibody, then it will be not getting bounded with this particular type of antigen. Now, after addition of the patient sample, this antibody gets bounded to this particular antigen, right? After this, there comes one washing step here, with the help of which, if there are again any unbounded antibodies present inside this solution, then they will get eluted out, they will get washed off, right, in this washing process. Then what do? You will be adding here secondary antibodies. Now these are the particular types of antibodies on which enzyme is linked here, and this particular type of antibody, if they found here an antibody is, or already an existing antibody is present here, then this antibody gets binded to this primary antibody, right? After this, what happens? Again, washing step is carried out. If there are some unbounded antibodies which are linked with enzyme, that will get washed off. And after that, what happens is, this particular, if there is a presence of enzyme, what is the next step? Obviously, there is a step, you are adding substrate, and after addition of substrate, this enzyme gets binded with the substrate to form the product, right? And with the help of which, as you can see, these are all light in color, these are all sky blue in color, this is dark blue in color. That means what? The product formation has been displayed the color inside this particular well. If the color develops inside a particular well, it means that there is a presence of specific type of antibody in the patient serum sample. This is your what? Indirect ELISA. Next, coming to the very next type, that is called as sandwich ELISA. Now, from the name itself, you can analyze. Sandwich means what? There are two layers of bread, and in like between them, what is there? There is a stuffing. Simultaneously, there are two antibodies like this, and in between there, what is there? There is the presence of antigen. With the help of this sandwich ELISA, you will be able to identify or detect a specific type of antigen is present inside a body or not. For that, first, inside this microtiter plate, what happens is, you are coating here what? Antibody. Since you want to detect the presence of antigen inside a patient sample, so for that, what you have to do, if you want this particular antigen to get binded with specific thing, that is what, that is the opposite of that. Means this is your thief, this is particular antigen, your thief which you want to find, and for that thief you are, what you are doing, already a soldier you are placing inside a well, right? This particular antibody is being coated inside the well, and after that what you are adding, the very second step, that is your addition of patient serum sample. In the very second step you are adding, antigen is added, means what, patient serum sample is added, through which if in the particular patient serum sample a specific antigen which is present for this particular specific antibody, then what happens is it gets attached or it forms a complex with the particular antibody, right? So here, after addition of the sample, if this antibody finds a specific antigen, then it gets bounded at its paratopes site, right? After this, again you are doing the washing process, and in this washing process what happens is, if there are some again antigens which are free in this particular solution, then they will get washed off. After washing off, what you are doing, you are adding the secondary antibodies, which are linked with enzyme, and after this what happens is, since there is the presence of enzyme, again washing step, right? For what, for excess amount of unbound type of like enzyme linked antibodies, they will get washed off, and after getting washed off you will add substrate, and this enzyme converts this substrate into a product. And as you can see from the screen, this is different color and rest of them are different color, that means color has been produced inside this well, and this color detects that there is a presence of specific type of antigen of interest, right? This is your third type of ELISA, which is called as sandwich ELISA. Coming to the last type of ELISA, which is called as competitive ELISA. From this method, you will be able to identify that how much amount of antigen is present inside a patient serum sample, right? From this method, we will be able to conduct, or we will be able to learn about competitive ELISA. What we are doing, firstly, you are taking the patient serum sample, and after taking this serum sample, what you are adding the antibody inside serum sample with the help of this micropipette. What you are doing here, after the addition of antibodies, you are making, or you are putting it for incubation duration, right? For this incubation duration, if on what type of antibodies, you are producing these antibodies outside, and after preparing these antibodies, which type of antibodies, which should be specific to this type of antigen, you are already preparing it in labs, and you are incubating or inoculating it inside this antigen, and with the help of which what happens, as you can see here, there will be bounding or complex formation between a specific type of antibody with the specific type of antigen, as well as you are adding a lot amount of antibodies, with the help of which what happens is, some unbound antibodies or free antibodies are also present inside this solution, right? Okay, so as we have seen already, these are your bound antibodies, right? First of all, what you are doing, you are adding bound primary antibodies which are already bound, and additionally you are also adding in this solution, which is termed as reference antigen, or you can also call it as inhibitor antigen, right? From the name itself, this is a type of competitive ELISA. So here in this particular type of competitive ELISA, there will be a competition between inhibitor antigen and patient's antigen. So this particular type of inhibitor antigen you are already preparing through lab, and patient's antigen, it is already present inside a patient. First of all, you have added bound first primary antibodies here, as you can see very clearly. Along with this, reference antigens are also present here. Next, as there is a presence of already primary antibody, and what is your next step? Your next step is addition of secondary antibodies. These secondary antibodies are linked with what? These are linked with enzyme, right? After addition of this, if they found some specific type of antibodies here, then this secondary antibody gets attached with primary antibody, and after getting attached with this particular primary antibody, what happens is, there is the addition of substrate. If the substrate gets bounded to this enzyme, then it will have the ability to produce a product, and with the help of product, what happens is, color occurs. from this image, we'll be able to clearly understand that what is the scenario going inside this particular competitive type of ELISA. Here you can clearly see that there are bounding, this complex formation of antigen, primary antibody and secondary antibody. There are two complexes, and in this particular diagram you can see there are four complexes. Since there are only presence of two complexes, means the color is lighter, and since there are the presence of four complexes, means that the color is darker as compared to this. With the help of this coloration, you will be able to identify whatever the antigens, what is happening here. There is, see, as you can see here, antigen is less and free antibody is more. As you can see here, this free antibody, at the very starting we have already said you in this image, you are adding ample amount of antibody, so that some are free and some are bounded. So what is happening here? See, there were some inhibitor antigens, right? What is happening here? This inhibitor antigen are as it is, means antibody concentration was less, so no any extra antibody was present here to get bounded with this antibody, so only two complexes were formed. In this scenario, what is happening? This was already present and there were free antibodies. As you can see here, free antibody is more, so these free antibodies are getting binded to this particular type of antigens, and after binding they will form a complex which will further produce colors with the like conversion of enzyme and substrate to the product formation, and this will liberate color. If there is a presence of darker color inside a particular well, it means that there are, what we are saying here, there are less amount of samples, your patient's antigen inside a solution, and from this particular light color, that there is less amount of antigen present inside this first well, and there is no any presence of patient serum sample, because what is happening here, this bounded and this free antibodies, that we are adding by ourselves. In the very this step we have discussed, we have added bounded and free antibodies. This free antibodies, what they have done, they get bounded to this remaining antibodies, with the help of which it has shown a darker color. So with the help of this, what you are clearly able to conclude from the lighter color, if antigen is more, free antibody is less, then in that condition you will be able to see a lighter color. If antigen is less and free antibody is more, so this free antibody, it gets bounded to this particular remaining antigens which are present there, and it has showed the darker color. So with the help of this, there was a competition between whom? Patient serum sample. This was your patient serum sample, this are your patient serum sample, and this is your inhibitor. So there was a competition of between these two, right? So with the help of coloration, this will be your control color, and if there arises a little lighter, that means some amount of antibody is present. Again, lighter color means more amount of antibody is present, right? So with the help of this, we have learned here 4 types of ELISA. So thank you so much for watching this video.